Hey, Aaron, can you hear me?
Okay, now can you hear me?
Yes, yeah, now I can hear you.
I okay. Do you want to test out your slides? I still can't hear you somehow.
What's that? I missed.
Okay. Can you hear me, Corey?
Yes,
Cool. Okay, yeah, I was just having some trouble with my audio. Um, can you test out your slides?
sure. I think, yeah,
Thanks for coming, by the way.
yeah. Thanks. Yeah, thanks for the invite, and thanks for your flexibility since I had to.
Oh, yeah.
Is
How long
that
have you been in San Francisco? Yeah, I can see it, but maybe try out the slideshow, the presentation.
okay? I forget, how do you do the presenter view thing on
Um,
Zoom? I don't know.
I don't know, try that, and then it might let you. Is that the presenter view or no?
No, let's
Okay. Yeah,
maybe that's it.
and then go to. Oh. Hmm.
How do you do that? No.
But then there's something there, yeah, usually there that says swap displays. So I don't.
And sharing. It doesn't matter. I'll remember my
Yeah, I have this new, or I don't know, new, but my Mac laptop, I can see on the bar on the top, like the next slide. That sometimes gives me a clue what's coming up next. All right. Hi, Shinjae.
Hi. Sorry,
Nice
I'm
to see you.
late, I guess. Nice to
No,
meet you.
perfect. Yeah, yeah, yeah. Thanks for coming. Can you test your slides
Okay.
just to see?
Just go to the
Yeah, looks
presentation
good.
mode.
Yeah, yeah.
That should be
Wow.
okay.
Okay, yeah.
I just came back from the Gordon conference. That should be good.
Oh, yeah. This is a little bit more intense than Gordon Conference, but I think it'll be good.
Oh, okay, sorry, stop sharing.
Okay, yeah. I used to live in Philadelphia.
Oh, okay.
Yeah,
Wow.
it's my
When
favorite place.
Bay
I
Area is my favorite place.
no, Bay Area is still like a fantasy world to me, but I was born in Philadelphia. I went to
Oh,
grad
okay.
school at college in Pennsylvania, grad school at UPenn. I worked at UPenn as an assistant professor
Oh, right,
growing
right,
up.
right,
Yeah,
right.
so I love it.
Oh, yeah.
Hi, Zilong.
Aaron, Shinjae, Corey,
Hi.
Hey,
nice to see you. Hey,
Aaron, I think you asked me how long I've been here. I forgot
Oh yeah,
to even
yeah, yeah.
answer the question. Messing around with my slides. So we got here in July.
Oh wow. Okay.
Yeah.
Nice. So actually, the February, March time, January, February, March is sort of like the best time in terms of comparison to the East Coast because it's really nice.
Yeah, although this is a weird time. It's like raining in April into May.
Yeah. Hopefully that'll stop.
but
Well,
where? Corey,
we
isn't
might need
that?
it in San Francisco.
Oh, okay.
Are you living in the city?
Yeah.
I live in Foster City.
Okay. So down.
Hmm.
Is that
That's
it's
like in San
north of
Mateo.
Palo Alto, right?
Yes. Yeah. Yeah.
Yeah.
Yeah. Yeah. South of the airport.
Yep.
Yeah. But right now I'm in Shanghai.
Oh, wow. Okay. I didn't realize.
Oh.
So is there still a lockdown in Shanghai? That's what I heard. A few
Yep.
weeks ago.
Yeah.
Okay.
Hopefully soon. We're expecting like in a couple of weeks. I don't know. See how the prediction
told-
works.
They
Yeah.
said like
Oh.
they said half the city 5 days, other half 5 days, but it's been what 3 weeks now.
Yeah, no, yeah, over a month. So we still have a fantasy it could be open up in a few weeks. I
Yeah.
don't know.
I went to the guard yesterday and I said, "Hey, can I just walk outside for 30 minutes?" He's like, "Bunang, no." It's like,
Yeah, you can walk your dogs, right? At least in the.
well, you're not. Well, I'm walking my dog, but you're not. You're not supposed to, but I have to.
Yeah, you know,
But just in the compound.
sections. Yeah.
Yeah, it's crazy.
So how long are you in Shanghai?
I've been here like almost a year.
Oh, are you
Yeah,
doing sabbatical or?
Sabbatical. Yeah.
Okay.
Yeah. I brought my family. My kids are going to school here.
I had a lot of party here already. So
Yeah.
nice.
It's crazy. Like we were. Yeah. Anyway, it was a good experience overall.
We're still hoping some summer party still, hopefully. Oh.
Yeah. I booked my ticket back in June 2nd.
Yeah, June, right? Okay.
Yeah, but maybe sooner.
So September still part of a summer in Shanghai. So yeah, still hot.
Sure. Yeah. So Corey, you're living in the city?
Yeah, so I'm currently in faculty housing, just down from Parnassus. So the lab is in the stem cell building on the Parnassus campus.
Okay, I know it. Yeah.
And we picked probably the worst time ever to try to renovate a house. So we have a house that's actually close to Twin Peaks.
Mm.
And
You bought a house?
So I lived
Wow.
here before as a postdoc.
Wow. And you bought a house as a postdoc?
Yeah. No, that's
wow. I
Like in
make
San
a ton of money.
Francisco.
I make a lot of money. I can't afford a house in San Francisco as a professor. Wow.
So
Okay, that's cool. Wow. Cool, but that'll be good to happen.
Hi, Yiming.
Hi.
Good to see you, Yiming.
Great.
Yeah, long time no see. Yeah.
Yeah. I'm here. Hi, Corey.
You San Diego?
Yes, in San Diego. Yes.
Sweet.
I
Hey,
texted
Yiming.
Emin. I said, Emin, there's a talk about hedgehog signaling and synapse development. He said, I'll be right there.
Absolutely have to be here. And for Corey, of course, any topic we have to be here, especially hedgehog.
Thanks.
Yeah.
Shinjae, do you know John Epstein at Penn?
Mhm. Yeah. We are in the same building. I'm a Smiler.
Yeah, so I was a grad student in his lab.
Oh, okay. See.
Uh-huh.
Yeah. I looked it up and then you're
Oh,
at
you looked
Penn.
it up. Okay. Yeah.
Yeah. And I looked at it a long time ago, so I just forgot about it.
Yeah.
Where was your lab?
BRB, 9th floor BRB.
Oh, BRB.
Yeah.
Okay.
And I used to eat. Do you know Jim Shorter? James Shorter.
Yeah. Yeah.
Yeah, I used to eat lunch with him every day at Abramson cafeteria.
I see. Yeah. I think we're close to Edmondson. Yeah. Smiler.
Okay. All right, so I can get started.
Sure. Go ahead. Yeah.
All right. Thanks, everyone. Another week of Neuro Zoom Shanghai lockdown edition continues. But we'll stay here, keep giving you good science to ease the pain and make us forget about our hunger and other things we're missing, but we have good science. So we have awesome talks today, and then I want to let you know that next week we'll have 2 more great talks. We have Renata Batista Brito from Albert Einstein, and we have Gang Cao from Huazhong Agricultural University. So tune in to hear those talks, and let me and email know when you're ready to present your work. We have lots of spots available throughout the summer, and 1 more talks in the spring. Okay, so it's my great pleasure to introduce our first speaker, Dr. Shinjae Chung. She's an assistant professor in the Department of Neuroscience at University of Pennsylvania. She received her undergraduate degree in Korea University, and then did her PhD at UC Irvine, where she studied the role of melanin concentrating hormone in modulating the reward in the dopaminergic system, and studied its effects on cocaine addiction. She then moved to UC Berkeley to do a postdoctoral fellowship with Professor Yang Dan, and here she, of course, studied sleep. And she had this amazing paper in Nature. I mean, it's amazing in and of itself, but this is actually a paper that I teach in my genetics for first year neuroscience students class at Stanford, because it has all of the techniques and all of the logic. It's really, I'm not just saying this, it really is an amazing paper. Started with the observation that in humans and other animals, if you lesion a certain region of the hypothalamus, it causes sleep problems. Other work had found there are certain neurons that are activated during sleep, but these were kind of mixed in with neurons that were inactivated. And she came up with this really extraordinary way of retrograde labeling the ones that are sleep active, not just stopping there, but then using ribosome profiling, using single cell sequencing to find all the markers. So basically, she's now captured control of these sleep active neurons in the circuitry. And amazing paper, and please read it, but please watch her talk to hear the latest. She still studies the mechanisms of sleep, but also how emotional regulation is also regulated. So looking forward to hearing your latest, Shinjae. Sorry, I'm excited to actually meet you because I've been talking about your paper for many years.
Yeah, thank you so much. Yeah, it's always great to hear that someone's reading my paper for many years. Okay, so do I have 20 minutes or 25 minutes?
20 to 25 minutes.
Okay, sounds good. Okay, yeah, thank you, Aaron, for the invitation. It's really a great pleasure to present our work here. So my lab is very interested in understanding neurocircuit mechanisms controlling sleep and how they become dysfunctional in neurodevelopmental and neuropsychiatric disorders. So today I'm going to talk about our work, the title of Noradrenergic Hypothalamic Neurosubstrate for Stress-Induced Sleep Disturbances. Okay. So we spend 1/3 of our lives in sleep. And as you can see here, all these animals spend a lot of time sleeping. And then we all experience when our sleep is bad, that this really disrupts our behavior and also our mental and physical health. And this video is from my son when he was just born, and he spent really a lot of time sleeping. And as you can see from his muscle twitches on his face, that sleep is incredibly important for neurodevelopment. So my lab is interested in understanding neurocircuit mechanisms underlying sleep and emotional regulation. We all experience that emotional stress disrupts our sleep, and when our sleep is bad, that negatively impacts our mental health. And this is from the New Yorker, the portraits of insomnia. So we all experience that when our sleep is bad, that we go to our bed and we just cannot fall asleep, as you can see here from this person, that he just cannot fall asleep, and that he stays awake all night thinking about something else, and then next day he just cannot function very well. It was also shown that the people who tend to sleep bad after stressful experiences tend to have insomnia or have an increased risk of developing other psychiatric disorders such as anxiety and depression. So we are very interested in understanding the neurocircuits underlying the impact of stress on sleep and which brain regions involved in stress regulation innervate sleep control circuit. So especially we focus on the locus coeruleus LC because this region has been shown to be important in regulating stress and arousal. So the LC contains this small class of neural cells, as you can see here, located in the pons. And the rats have about 1,500 cells on each side of their brains. And LC projects widely to the entire forebrain, brainstem, and cerebellum. And LC is implicated in regulating attention, learning and memory, and decision making. And LC is also implicated in neuropsychiatric disorders through its role in regulating stress responses. So the role of LC in regulating arousal have been very well documented in the past decades. So Gary and Sir Jones published this paper recording LC neurodivergent neurons, as you can see here. And he showed that the LC neurodivergent neurons are most active during wake and less active during non-REM sleep. As you can see here in the single unit activity, and it's almost silent during REM sleep. And the mean framing rate is highest during wake and lower during slow wave sleep and almost silent during REM sleep. And also Louis-Dill's group also published that the optogenetic stimulation of LC neurons promotes arousal, as you can see here from this Chung group. So our lab was interested in understanding how LC neurons, their activity changes during sleep and wakefulness after stress. So here, first, we wanted to record LC activity during spontaneous sleep and wakefulness. So here we decided to perform the fiber photometry experiment by using DBH pre-mice and injected AAV containing GCAN6S, which is a calcium indicator, into the LC and implanted optic fiber to perform fiber photometry. And this figure shows the LC cells, neural cells expressing GCAN6S. And here is a fiber location to image the population activity of LC neurons. And this is how the LC activity looks like. So here is an example recording. Here is EEG, EMG, color-coded brain state, and the activity of LC neurons. So as expected, the LC neurons are most active when the mouse is awake, as shown here in this purple, and less active during non-REM sleep and least active during REM sleep. But we found it very interesting that it's not that LC neurons are just less active during non-REM sleep, but rather the LC neurons become rhythmically activated during non-REM sleep. So we wanted to understand what kind of rhythm the LC neurons are following during non-REM sleep. So previously, it was shown that the EEG sigma power fluctuate on a minute time scale, which is called infraslow rhythm. So the sleep spindles are the major contributor to the EEG sigma power. That's about 10 to 15 hertz. And depending on the number of sleep spindles during non-REM sleep, shown here, you can see here that if there are a lot of sleep spindles occur, then this increases EEG sigma power, as you can see here. So the Untitled group look at the EEG sigma power fluctuation in mice and human. Here they look at the power spectral density of EEG sigma power, and they found a peak at around 0.02 hertz, which means that this fluctuation happens at about minute time scale. And it was also similar for the human. So we wonder whether LC neurons actually follow this infraslow rhythm during non-REM sleep. So this is how the recurrent looks like, and during non-REM sleep, they're activated. So the activity really fluctuates, and then we zoom into this region in the non-REM sleep. And here is the EEG sigma power shown in green, you can see here. And this is the activity of LC neurons. And whenever the sigma power is high, the LC tends to be silent. But whenever the sigma power goes down, the LC neurons become activated. And the sigma power goes up again and goes down, and the LC neurons become activated. So we also look at the normalized power spectral density of this EEG sigma power shown in green and the LC activity shown in blue. And then when we plot this, we found also the peak at a similar area, somewhere around the 0.02 hertz, which means that this fluctuation also happened at about minute time scale. When the EEG sigma power goes high from trough to peak, then the LC activity goes down. To understand how the LC activity fluctuates during non-REM sleep, we also look at the cross-correlation of LC activity, delta F over F, with the EEG, delta, theta, and sigma power, and found that they are negatively correlated, suggesting that the EEG sigma power and LC activity exhibit infraslow oscillation, but they are negatively correlated. So whenever the sigma power in the EEG goes high, the LC becomes silent, but when the sigma power goes down, the LC becomes activated. And then we also found something else that's interesting, that whenever the LC becomes activated, that the EEG become desynchronized and EMG shows brief muscle activation. We call this as microarousals. So what are microarousals? So the microarousal is a regular phenomenon of our nocturnal sleep. So all of us have it. For example, that when we are sleeping, that sometimes we briefly wake up and then we fall back to sleep. So that's called the arousals lived into sleep. And the EEG and polygraphic characteristics of transient shifts toward arousals. However, when we have too many microarousals during our sleep, that has been shown to be involved in the pathophysiology of sleep disorders. And this figure shows how EEG and EMG looks like during wake, microarousals, and non-REM sleep. So during wakefulness, EEG is desynchronized, EMG shows muscle activation. During microarousals, the EEG is desynchronized, and EMG also shows brief muscle activation. We also look at the EEG delta power to see how microarousals are different compared to non-REM sleep or wakefulness. So during non-REM sleep, the EEG delta power is high, reflecting the slow oscillation in the EEG. However, during microarousal, the EEG delta power is lower, but that was actually higher when the mouse was awake. For the EMG power during microarousals was also in between the wakefulness and non-REM sleep. So from here, we could conclude that the microarousals are a distinct brain state that's different from non-REM sleep or wakefulness. So we also look at when LC activity changes compared to the microarousals. And then here we plot this LC activity, delta F over F, and this is time relative to microarousal onset. So this pink line indicates when the microarousal happened. And as you can see here, the LC activity increased about 1 second before the microarousal onset. So LC activity increased before about 1 second before the microarousals. So we wonder how stress impacts the sleep microarchitecture and the LC activity during sleep. So here we used an acute social distress paradigm. So our experimental mouse was exposed to the acute social distress, where the experimental mouse was actually defeated by this aggressive CD1 mouse. And then we separate them by the perforated wall, and then we perform the sleep recording while recording the LC activity. We first look at the overall time spent on sleep and wakefulness. And as expected, our experimental mice spend more time awake and then less time on non-REM and REM sleep. And then we also look at the changes in non-REM sleep microarchitecture. For example, the microarousals were shown to be increased after stress. We also look at the number of sleep spindles, and that was significantly decreased after stress. We also look at the EEG sigma power fluctuation to look at the infraslow rhythm, and gray line indicates a baseline, and this blue line indicates a stress. And we found that after stress, the infraslow rhythm was also disrupted. We also look at the strength of this fluctuation. Strength means the area under this curve. And we found that compared to the baseline shown here in this gray line, showing a really high peak at around 0.02 hertz, that after stress, the sigma power fluctuation was significantly disrupted. So from here, we could conclude that non-REM sleep is fragmented by increased microarousals after stress. And also the number of sleep spindles is decreased, and the infraslow fluctuation of the EEG sigma power is impaired. And all these changes during non-REM sleep also impact the transition to REM sleep, and then they actually tend to transition less to REM sleep. So we wanted to look at how LC activity changes after stress. So here is from the baseline recording. So here is EEG, EMG, and the color-coded brain state, and the activity of LC neurons. And from time to time, the LC becomes activated during non-REM sleep, and sometimes they overlap with microarousals during sleep. And this is how it looks like after stress. So here is LC activity, and then here is color-coded brain state. As you can see here, the LC becomes really more frequently activated during non-REM sleep, and they also tend to overlap more with microarousals, which means that their sleep is also fragmented. So we counted the number of calcium transients during non-REM sleep, and here, as you can see here, this gray bar is from baseline, and this is after stress. So the stress increased the number of calcium transients, which means that those are actually the calcium peaks. So LC tend to become more activated during non-REM sleep. We also look at the probability of the calcium transients overlapping with microarousals, and also found that probability was also increased, which means that there are more LC activation during non-REM sleep, and they tend to overlap more with microarousals. We also look at the infraslow modulation of LC activity, and then here is a normalized power spectral density during baseline recording shown in here in gray, and after stress, that's in blue. We also look at the strength of this infraslow fluctuation that was also disrupted after stress. So from here, the stress increased LC calcium transients and disrupt the infraslow modulation of LC activity during non-REM sleep. So we wonder whether LC activity is sufficient to induce microarousals. So here we performed optogenic stimulation experiment by stimulating LC to see whether we can actually induce microarousal whenever we stimulate them. So here we use DBH pre-mice and injected AAV contained channelopsin into the LC and the implanted optic fiber to be able to stimulate those LC neurons. So here is an example recording. So we stimulate LC neurons from time to time, every 15 or 25 minutes for about 20 seconds. And here, when we stimulate the LC neurons, it promotes microarousal shown in pink, and also here, and also here. And this is a zoom in figure for this period. Here is EEG trace, and here is EMG trace. So when we stimulate LC neurons, that we found that this was able to trigger microarousals characterized by desynchronous EEG with muscle activation. But we want to look whether this is actually similar to the spontaneous microarousals. So we actually zoom in into this microarousal, as you can see here. And the EEG and EMG signature look also very similar compared to the laser-triggered microarousals. But we actually wonder whether the reason that we induce microarousal is because we stimulate LC neurons only for 20 seconds. So we decide to stimulate longer period of time, such as like 120 seconds, to see whether we can actually induce long wake bouts instead of short microarousals. So here we stimulate LC neurons for 5 seconds, 20 seconds, or 120 seconds, and then look at the arousal duration to see whether when we stimulate longer period of time, whether we can actually induce long wake bouts. And here is our arousal duration, and this is probability on the y-axis. And then we found that regardless of the laser stimulation duration, such as 5, 20, 120 seconds, that it actually shows a peak at around 5 seconds, which means that regardless of the laser stimulation duration, that we could only induce microarousals without inducing long wake bouts. So then we wonder whether LC activity after stress is really necessary for stress-induced microarousals or the stress-induced sleep disturbances. So to do this, we decide to inhibit LC neurons by using chemogenetics. So here in the DBH pre-mice, we injected AAV containing HM4DGI and cherry, or AAV containing N cherry. So during baseline recordings, we injected the CNO into 2 groups and then performed sleep recording to see how the CNO injection can change their sleep. And also during stress experiments, the mice were exposed to acute social distress, followed by CNO injection. And then we performed sleep recording to understand how LC inhibition could impact the stress-induced sleep disturbances. And this is how the M-cherry mice look like after stress exposure. And this is HM4DGI, so LC inhibition group look like after stress. So you can notice this sleep fragmentation induced by microarousals. And after, when we actually inhibit LC neurons, after stress, that actually the number of microarousals was decreased. And here is actually a summary to show you the microarousals. So this is during the baseline recordings, and this is from M-cherry group, and this is HM4DGI. So inhibition of LC neurons actually decreased the number of microarousals. And after stress, the microarousal was increased from here to here, from the baseline to stress. But when we inhibit LC neurons, that was significantly disrupted. Significantly decreased, suggesting that the sleep fragmentation was actually reversed when we actually blocked the LC activity. So we also look at the number of sleep spindles. So that was not changed after when we actually block LC activity during baseline, but the sleep spindle, the number was decreased when the mouse was stressed, and that was actually reversed when we blocked the LC activity. So interestingly, the amount of time spent on awake non-REM or REM sleep was not changing after LC inhibition, suggesting that the LC activation after stress may contribute to the sleep fragmentation, but not necessarily the amount of time spent on sleep. But then we wonder whether LC neurons actually regulate the sleep microarchitecture by interacting with the core sleep control circuit. And then previously was shown that the noradrenaline suppressed the putative sleep cells in the preoptic area. And those putative sleep cells are called the low threshold spike, LTS cells. And so what they did in this paper in 2000 by developing that they actually used a brain slice containing POA, and that they applied the noradrenaline, and they found that these LTS cells the activity was suppressed, whereas the non-LTS cells were excited when they applied noradrenaline. However, this actual study we cannot really figure out whether these putative sleep cells are really active during sleep in vivo. And also when we look at the LC fibers where they project to in the brain, we also found that the LC neuroenergic fibers in the preoptic area, suggesting that the LC neurons can really innervate the preoptic area to regulate the activity of sleep or wake cells in the region. Then here we wanted to look at whether norepinephrine is released in the preoptic area during sleep. So for this, they use a genetically encoded fluorescence sensor for norepinephrine detection. To measure norepinephrine level in the preoptic area, we injected this AAV graph sensor into the preoptic area and implanted an optic fiber to the preoptic area to be able to measure the level or the calcium activity, the fluorescence. And here is an example recording. So here's EEG, EMG, color-coded brain state, and this delta F over. So this represents the norepinephrine level in the preoptic area during sleep and wakefulness. And as expected during wake, that the level was highest, and during non-REM sleep, it was also there, and they also show some fluctuation during non-REM sleep. And then when the mouse was in REM sleep, that was actually really suppressed. So we zoom into this non-REM period to see how that really correlates to this EEG sigma power fluctuation. And here is the EEG sigma power fluctuation shown in green. And then this is activity. So this is activity or delta F over F. And as you can see here also that when the sigma power goes up and down, that this calcium activity tends to increase, suggesting that they also show this calcium activity shows also infraslow rhythm, and they are negatively correlate with the EEG sigma power. And then we also wonder whether these neuroenergic fibers in the preoptic area can change the brain state. So here we injected AAV containing channelopsin into the DBH cream mice, and then implanted optic fiber to the preoptic area so that we can actually stimulate these projection fibers. And then we shine the laser from time to time and found that the stimulating these projecting fibers that can also promote microarousal. As you can see here, this is a laser trigger microarousals, and the EEG and EMG shows that desynchronous EEG and the EMG muscle activation, which was similar to the spontaneous microarousals. So stimulating LC projections to the preoptic area promotes brief arousals. And then we wonder how these neuroenergic fibers coming from the LC change the activity of preoptic area neurons. So the preoptic area was shown to contain a variety of neurons that I actually characterized in my paper that was published back in 2017. So preoptic area contains a lot of REM active neurons. About 50% of neurons are most active during REM sleep. And these REM sleep active neurons could be further divided into REM, non-REM active neurons, or REM and wake active neurons, and I'm going to talk about that later. And about 10% of neurons are most active during non-REM sleep, and about a quarter of them is most active during wakefulness. And then we wonder how these neurons actually change activity when we actually stimulate these neuroenergic fibers in the preoptic area. So to investigate that, we use some optical recordings. So optical consists of conventional microelectrodes glued to an optic fiber so that we can stimulate these fibers in the preoptic area while measuring the activity of the preoptic area neurons. And this is a summary of all the neurons that we recorded from the preoptic area. So here, the blue cells are the ones that are inhibited by the laser stimulation, and these orange cells are the ones that are excited by the laser stimulation. And the black dots are the ones that are not responsive to the laser stimulation. And we put all these dots in this quadrant. So the x-axis represents the relative activity for non-REM versus wake. So if they're more active in non-REM, they are located on the right side. If they're more active during wake, they're located on the left. And the y-axis represents the activity for REM versus wake. If they're more active during REM, they're located on the top. So the cells that are inhibited by this laser stimulation are more located on this top right side corner, suggesting they're more active during REM and non-REM sleep. On the other hand, the excited neurons are located on the left, which means that they're more active during REM and wakefulness. And here is a summary of the preoptic area neurons that are inhibited by this LC-POA fiber stimulation. And here is a raster plot of this example cell. So when we shine the laser, the firing rate was significantly suppressed. And in total, we recorded from 45 cells. And about 20 cells were most active during REM and non-REM sleep, and 11 of them were most active during non-REM sleep. And here's an example recording from this REM and non-REM active neuron. As you can see here in this green line that represents the firing rate of this cell. This cell is most active during REM sleep, as shown here, and then here in this blue spot. But when the mouse was awake, shown here in this purple, is relatively less active. And during non-REM sleep, it shows also some fluctuating activity that whenever the mouse enters microarousals, that these cells become less active. So here, the average firing rate was highest during REM sleep, followed by non-REM and wake. And we also wanted to look at how the activity fluctuates during non-REM sleep. And so we look at the cross correlation of their firing rates with EEG, delta, theta, and sigma power. And then we found that their activity, the firing rates are positively correlate with EEG, theta, and sigma power, but not with delta power. We also found that their firing rates are significantly increased during sleep spindles, and their activity was suppressed when there are microarousals. And here's a summary from the preoptic area neurons that are activated by LC2POA fiber stimulation. And this is an example cell. So whenever we shine the laser, as you can see here, the firing rate was increased. And in total, we recorded from 17 cells. And about 8 cells were most active during wake, and the 6 cells were most active during REM and wake, which was different from the REM and non-REM cells that are suppressed by the laser stimulation. And here's an example recording when the mouse was awake. As you can see here, this cell was most active and was least active during REM sleep. And during non-REM sleep, they also from time to time become activated. And also they tend to overlap with microarousal shown here in this pink. And then we also look at the cross correlation of their firing rate with EEG, delta, theta, and sigma power to look at how the activity during non-REM sleep fluctuate. And found that their activity is negatively correlated with EEG, theta, and sigma power. And also during non-REM sleep, whenever there are microarousals, their activity was significantly increased. So by using optical recording, we really cannot figure out their cell types. So we can actually record that they are sleep active, but we actually don't know their cell type, that they are GABAergic neurons or glutamatergic neurons. So here, we wanted to use a photometry recording to really understand, to record from GABAergic neurons or different cell types or the glutamatergic neurons to see when they become activated. So here is a summary of the GABAergic neurons. And so here is an example recording. So whenever the mouse enters REM sleep, shown here and then here, the GABAergic neurons are most active during REM sleep. And here is an example recording from CCK neurons, which is a GABAergic subtype. And this cell was also most active during REM sleep. You can see here. And we also performed the microendoscope imaging to show that, so these cells are the CCK neurons in the preoptic area that we see from the green lens. And now the mouse is entering from non-REM to REM sleep, and the CCK neurons will actually become activated during REM sleep, suggesting these cells are sleep active. And then we also recorded from glutamatergic neurons, and these cells are most active during wakefulness, as shown here and also here, and then least active during REM sleep. And also during non-REM sleep, they also show some activation, and then also their activation tend to overlap with microarousals. So in summary, I showed that the LC neurons become rhythmically activated during non-REM sleep, following the Infraslow rhythm. and they are negatively correlated with the infraslow rhythm in the EEG. And whenever the LC activity is high, that they tend to overlap with the microarousals. But when the LC activity is low, the mice really have a higher chance to enter REM sleep. And when we activate LC neurons that actually promote microarousal, resulting in sleep fragmentation. And when the mouse was stressed, that the LC neurons become more activated and they tend to overlap with more microarousals. And the LC neurons cannot be really silenced enough that the mouse really cannot enter REM sleep. And these changes really may contribute to their to impair sleep quality after stress. And we also show that the LC neurons project to the preoptic area, and they inhibit the sleep neurons, especially REM and non-REM active neurons, while exciting wake and REM active neurons. So yes, I'm on time. I think, okay. So
Thanks.
Finish my talk. Yeah, by thanking everyone in my lab. Yeah, thank you so much. I will take any questions.
Great, thanks. Thanks so much. Awesome talk. There's a question from Lin. Do you want to ask it?
Yes, great. Yeah, Aaron, can you hear me?
Yes.
I can hear you, yeah.
Okay. I might have missed, but can you explain the microarousals? How do you identify them? Because I saw these EMG recording the muscle twitches, but they also can be observed when the mice are resting awake. So I wonder, yeah, the differences between these muscle twitches.
Oh, so during sleep. Oops.
Yes. Also, yes.
Oh, yeah.
So I wonder how to identify these microarousals. I kind of missed like the definition of the microarousal. And then,
oh, I see.
Yeah. And then another question is, when you activate the LCNE activation, would you see more of the non-REM to wake transition or non-REM to REM transition? Yeah.
Okay, so I will just first go over this microarousals. You can see this. Yeah, so okay, so the microarousals. So basically, this happens. The microarousal happens during sleep. So during sleep, that we actually look at their EEG signature. So this basically it looks like this. So EEG looks like desynchronized, similar to wake, but probably not as intense. And then EMG shows a brief muscle activation. But it's less than the wake bout, less than 20 seconds. So yeah, so this actually microarousal happen during non-REM sleep. So during like quiet wake, because during wakefulness, we actually don't search for microarousals, but whatever the duration is less than 20 seconds, we actually classify them as microarousals. Does it make sense? Did I answer your question?
Yes. So
and then what was the second?
That is like the 20 microsecond muscle twitches during non-REM sleep.
Right.
So do you see any of these in REM sleep?
So we didn't actually look at that because it doesn't seem to be really, the mice just tend to wake up during REM sleep, but we actually didn't see much about the microarousals during REM sleep. It just seems to be more the non-REM sleep phenomenon.
I see.
And also, we also look at the infraslow rhythm during non-REM sleep. So.
Okay, thanks.
What was the second question? Actually, the non-REM.
The LC, yeah, the transition. If you activate the LC to NE neurons, will that trigger the non-REM directly to wake, or it's non-REM to REM?
When we stimulate LC neurons.
I remember you had LCNE activation.
Yeah. So if you stimulate LC neurons, they will induce wakefulness, not REM sleep.
Oh, okay. So it's directly to wake.
Right. The microarousals, yeah.
Okay, thanks.
So, Shinjae, I'm just wondering, these microarousals, are they just incidental, or are they sort of part of the sleep program? I just noticed when you silenced the locus coeruleus neurons in the baseline, it seemed like there were a little bit fewer microarousals. And I'm just wondering, like, what's the consequence of an animal having fewer than normal microarousals?
Yeah, so the microarousal, yeah, it kind of looks random, but then we also found that the microarousal is really part of the sleep architecture. So for example, the mouse, so when the mouse sleep is really bad, for example, after stress, the mouse really cannot enter REM sleep, for example. And then microarousals tend to happen more, or no, they tend to happen less before they transition into REM sleep, for example. So the microarousal is kind of part of the sleep architecture that when there are too many microarousals that really disrupt the whole sleep architecture, such as transitioning into REM sleep, for example.
Got it. Okay, any other questions? Okay, if
Hey,
others come
Shinjae.
up. Oh, okay, Kaiwen, go ahead.
Yes, yeah. Hi, Shinjae. Great talk. So I have a questions about the inhibition of the alpha-synergic neurons after stress, with or without stress. So it seems like it doesn't really change the percentage of sleep-wake stages. So I'm just curious whether you measured for the 24 hours app or just during just a very short, brief time of recording.
Yeah, I mean, that's a great question. So we only measure 4 hours, but it's possible that maybe it changes afterwards. But we also use thread. So the Sienna doesn't last that long.
Okay.
But,
okay.
Yeah, but we only look at 4
that
hours.
is okay. 4 hours post stress.
Right.
Okay. Great. Okay. Thank you.
Okay, thanks so much, Shinjae. And if others have questions, you can put them in the chat, and I'm sure Shinjae can answer. Zilong, you're up.
Okay, great. It's my great pleasure to introduce the Dr. Corey Harwell. He's from now an associate professor in UCSF. So Corey has his PhD with NELC-POA in MIT, where he studied activity-dependent expression. So after the PhD, he went to West Coast, worked with Arnold Crexton in UCSF, studied the cortical development, based on early stage of cortical development. So after that, Corey stayed in the UCSF, carry on to study further, has a beautiful paper and regarding to the epigenetic relation, as well as the very basic classic morphogen, like solid hedgehog signaling in cortical synapse development. So today, Corey will share with his latest hedgehog signaling from neuron to astrocyte during the cortical synapse development. So welcome, Corey. Take it away. Yeah, we can see your slide. Go ahead.
Yeah, sorry, it's just unmuting. Thank you. Yeah, thank you for the invitation. So actually, I'm going to talk a bit about some work that started actually my postdoc at UCSF. And it actually began with this observation that we had that it's just mentioned that sonic hedgehog, the secreted morphogen, is expressed in the postnatal developing brain. And we were specifically interested in this pattern of a sonic hedgehog expression in the cortex. So I'm just showing images from sonic Cree-ER mouse, in which we administered tamoxifen between P8 and P9, and then harvested the brains at P28. And you can see this very distinctive pattern of fluorescent labeled cells that are in layer 5 of the cortex. And we further characterized those cells and found that those layer 5 neurons are nearly exclusively the subcortical projection neurons found in the cortex. But then we're not going to discuss here, but you can see in the image that there's actually patterns of sonic hedgehog expression across other populations of neurons in the postnatal brain, including areas like the claustrum, the thalamus, globus pallidus, and the hypothalamus. So just to kind of recap some of the previous work that we did. So we followed up on this question of, what exactly the sonic hedgehog expressed in the cortex is doing. And it was expressed during this period that coincides with the peak period of cortical synaptogenesis, between first and second postnatal weeks of cortical development in the mouse. And what we found is that there was expression of a receptor for sonic, a less well-known receptor called Bach, which is a part of the Ig superfamily that's expressed and a very complementary population of neurons. So Bach was exclusively expressed in callosal projection neurons within the cortex, so primarily in the more superficial layers of the cortex, while the sonic was expressed in these deep layer neurons. And using a series of genetic approaches, we found that either the loss of the Bach or sonic hedgehog would result in a reduction of the connections, the functional connections between these superficial and deep layer neurons, so layer 2, 3 to layer 5. So when I was setting up my lab subsequently, one way that we were following up on this is that we were just curious as to whether there were other components of the hedgehog signaling pathway that are expressed also in the postnatal cortex, and whether they had either cell type or layer specific patterns of expression. And one of the first receptors that we looked at is the more well-known receptor for sonic hedgehog, actually, patch1. And patch is indeed expressed in the cortex. So these are just images from developing postnatal cortex going from P5 to P60. And we first noticed the expression starting right at P5, where these beta-gal cells in this patch-laxi mouse are primarily distributed in the more deep parts of the cortex. But then by P15, you can see that the distribution of patch expression is throughout the cortical layers. And one difference I should point out with what we observe with patch expression compared to sonic hedgehog. So sonic is developmentally expressed in that it reaches this peak period of expression between those first and second postnatal weeks, and then it's rapidly downregulated in those neurons. Where with patch, what we see is that once it's turned on in those cells, it continues to be expressed throughout the life of the animals. When we look at the laminar pattern of enrichment of sonic, as I mentioned, that it first comes on in these more deep layers in the early periods of development, but then you see this more even distribution as the animals develop. And we further characterize the cell type specific expression of patch by looking at markers for different cell types, including neurons, microglia, OLIG2, either oligo precursors or glial precursors, depending on the age, and then of course astrocytes. And what we found is that at P5, nearly all of the beta-gal positive cells were actually positive for OLIG2. And we think that what they're actually labeling is the glial precursors. And it's actually quite striking that when you look at P15, there's almost a complete switch where there's almost, where there's very few OLIG2 beta-gal positive cells, and nearly all of the cells at that point are positive for S100 beta. And that continues through the second postnatal week, even up through the first month. And then at P60, we made this really interesting observation that I won't talk about much here, is that we start to see a subpopulation of neurons that is co-labeled with beta-gal. So that's potentially expressing patch at these late postnatal age. So if we know from, I guess probably now, 40 years of study of the sonic hedgehog signaling pathway, we know that patch is the one of the kind of primary receptors for sonic hedgehog, and the main function of patch in the hedgehog signaling pathway is to repress the activating receptor smoothen. And it's only upon the binding of sonic hedgehog to patch, in which the repression of smoothen is released, which then leads to the activation of these downstream Gli family transcription factors. And we know from work from many other groups, including Denise Garcia, when she was in Alex Joyner's lab, that GLI transcription factors are actually exclusively expressed in astrocytes in the cortex. And we actually did this collaborative experiment with Denise's lab, in which she took the GLI1 Cree-ER mice that they had been using and administered tamoxifen during that same first postnatal week, alongside our own experiments where we administered tamoxifen in the sonic Cree-ER mice. And what you can see is that there's this overlapping pattern of where the sonic hedgehog is enriched in the deep layers of the cortex, with the enriched numbers of these GLI1 positive astrocytes, suggesting the signaling is taking place around the somatic dendritic regions of those deep layer cortical astrocytes. So this gets to, our kind of the question that we have all of this evidence that there's signaling taking place, but what is it that the neurons might actually be telling these astrocytes to do during cortical development? And of course, there's a host of functions that are mediated by astrocytes, where they have important roles in ion homeostasis, neurotransmitter uptake, and during development, even sculpting the number of synapses. So one of our first steps in trying to address, how is it that sonic hedgehog signaling is impacting astrocytes, is that we went back to our sonic hedgehog conditional knockouts, and then specifically labeled populations of astrocytes using an approach called just using postnatal electroporation, using this technique called PALE. And that allows us to sparsely label groups of astrocytes in both the wild type and sonic hedgehog conditional knockout mice. And what we found is that there's a specific reduction in the complexity of the morphology of the astrocytes in the deep layers of the cortex. So again, in proximity to the source of those sonic hedgehog expressing cells, where we don't see that subsequent change when we look at the morphology of upper layer astrocytes in the knockout. And we looked actually at the ultrastructure of the astrocytes to look at their interactions with synapses by using electron microscopy in a collaborative set of experiments with Wei-Chung Lee's lab at Boston Children's at Harvard. And here we also see this specific reduction in the extent of the ensheathment or envelopment of neuronal synapses in the sonic hedgehog knockouts. I should point out that this observation seems to be, we see it in both the superficial and the deep layers of the cortex, where there's reduction in astrocyte ensheathment of synapses. So, so far we found that this neuron-derived sonic is required for the morphological complexity of deep layer astrocytes and its interaction with synapses, which has potential implications into how those astrocytes might sample the activity of the neurons around them and maintain homeostasis. But what are the molecular targets that these that sonic signaling in these astrocytes are actually driving? So to do this, we use the molecular genetic approach, where we sequence the astrocytes in which we could then manipulate sonic hedgehog signaling. So it's an outline of the experiment where we used our patch knockout. So it's important to note that, as I mentioned, patch is, in the absence of hedgehog binding, is a repressor of hedgehog signaling. So it's the obligate repressor. So the function of knocking out patch actually allows hedgehog signaling to be activated independent of hedgehog binding. So in effect, it produces a constitutive activation upon the administration of tamoxifen. So we crossed our patch conditional allele to glass-Crete ER and administered tamoxifen between P12 and P14. And we also included a Cree reporter so that we could sort out those recombined astrocytes and then subsequently sequence them. So we found that there's a number of genes that were differentially expressed. It's about 100 or nearly 200 genes that are differentially expressed using a log2 fold change greater than 1. But what we noticed is that if we look at those genes that were differentially expressed, many of them had known roles in regulating synapse formation from astrocytes. So for instance, this gene SPARC, KCNJ16, which encodes this inward rectifying potassium channel, LRIG1 had previously in neurons been shown to regulate neuronal morphology, and IL-33 is the secreted interleukin that had previously been shown to have a role in regulating synaptic pruning. So we were able to validate the expression of many of these genes in situ by either using antibodies. So here's an example with KR4.1 and LRIG1. And should point out that for both of these genes, they actually, in addition to being specific to astrocytes, they also have a laminar specific pattern of enrichment, where KR4.1, as you can see, is more highly enriched in layer 4 and layer 5B astrocytes. And when we look in the patch knockout, you see that there's actually an increase in KR4.1 and LRIG1 expression across all of the layers of the cortex. Again, suggesting that even though these genes have layer-specific patterns of enrichment, that the astrocytes have the capacity to activate the expression of these subsets of genes across all layers of the cortex. And here's just examples of a few of the other synapse-related genes that I mentioned, including IL-33 and SPARC. IL-33 is, I just mentioned that this increase that we see is both for IL-33, we can see the increase in the numbers of IL-33 positive cells, whereas for genes like SPARC, we can also see that there's increase in both the number of SPARC expressing cells and the intensity of the expression. So, so far we've shown that, activating the hedgehog pathway through our loss of patch function induces an increase in these synapse-related genes. So we essentially did the converse experiment in our sonic hedgehog conditional knockout to see whether we lose expression of those same sonic target genes. And we observed that. So here's just one set of examples, but this is consistent for all 4 of the genes that I've mentioned previously, is that if you look at IL-33, you can see that there's actually a really drastic reduction in IL-33 expression in the developing cortex in the sonic hedgehog conditional knockout. And the same we observe with SPARC. It's worth mentioning that these reductions in these genes is seen not just in the deep layers, but actually you can see the reduction throughout all of the layers in the cortex, even though IL-33 is most heavily enriched, say in layer 5, you can see that the reduction spans across all cortical layers. So, we found that the sonic hedgehog signaling influences these astrocyte-specific transcriptional programs and especially for genes that are known to be associated with synapse formation and function. So finally, we wanted to ask is, does promoting hedgehog signaling in astrocytes function to promote synapse formation? So to do this, we actually went back to our patch knockout and looked at the number of synapses around the territories of the individual astrocytes. So of the recombined astrocytes, is there a change in synapses? And we counted the number of V-GLUT PSD 95 positive synapses. And we see an increase of both V-GLUT1 and V-GLUT2 positive synapses in both the superficial and deep layers of the cortex. And we further tested whether this increase in the number of structural synapses corresponds to increases in functional connectivity in a series of physiology experiments. We looked at both just the mini EPSC frequency, which was increased in the patch knockouts, but then also we did a variation of a previous experiment we'd done in the sonic hedgehog knockout, but this time just using electrical stimulation, where we stimulate neurons in layer 2-3 and then record their postsynaptic responses or postsynaptic currents of neurons located in layer 5. And we see this significant increase in the amplitudes of the EPSCs upon stimulation in layer 2. So, so far we've seen that modulation of hedgehog signaling in astrocytes impacts both their morphology and their effects on synapse formation. So we wondered whether we could identify specific sonic hedgehog target genes that are involved in regulating these distinctive cellular processes. So we decided to take a approach where we would knock down several of the sonic hedgehog target genes, including LRIG, IL-33, and SPARC, and then assess the morphology of the astrocytes upon this knockdown. And what we found is that the knockdown of LRIG1 leads to a reduction in the morphological complexity of those cortical astrocytes. While we did the same experiment, but this time looking at synapses, and again looking at LRIG1, SPARC, and IL-33. And this time we see that there's no change in the number of synapses in the astrocyte territory in LRIG1, but there's a significant reduction in synapse density upon the SPARC knockdown. And this reduction in synapses is specific to V-GLUT1 synapses, while we don't see a reduction in V-GLUT2 inputs when in the SPARC knockdown. So just to kind of conclude, what we found is that expression of the sonic hedgehog receptor patch is exclusive to astrocytes during cortical development, where we then see this neuronal expression at a much later stage in the animal's development. And that sonic is both sufficient and necessary for regulating this layer-enriched gene expression patterns of astrocytes in the cortex. And then it's required for both the astrocyte morphogenesis and envelopment of those synapses. And importantly, we showed that we could actually resolve the distinctive contributions of different sonic hedgehog target genes to either astrocyte morphogenesis or towards synapse formation with SPARC. So I just will quickly acknowledge all of the current and past members of the lab who contributed to this project and our collaborators, especially Wei-Chung Lee's lab, who helped us, really generously helped us with the EM, and Michael Tolstoy-Rukov, who helped us with a lot of our RNA-seq analysis, and the Sabatini lab with the electrophysiology. And thank you for your attention.
Great, that's great work, beautiful. So now the open for questions. So may I ask the first one, Corey? So it's amazing. So how the hedgehog signaling is astrocytes. So I'm thinking about, so is that possible to do the classic assay like Ben Barres did, like separate neuron like glial in culture and to manipulate the patch 1 in the astrocyte and then see how this synapse formation in culture works. That might give you a can separate the full pathway. Is that,
yeah, absolutely. So you mean to see whether we can isolate the specific factors that are downstream of the sonic activation? Yeah, I think
that's just beside that. So I'm hoping so in vivo we show signal, but it's very complicated. So I saw the classic experiment Ben Barres did. It's in culture, right? So you separate astrocytes and separate neuron and put in the co-culture system, and where you can more clearly knocking out patch 1 or the downstream molecules. So how to see how the precisely its synaptic development compromised or increased in culture.
I see.
I don't know. It's not possible to.
Yeah, I think that this approach, because we have genetic handles where we can specifically manipulate both the neuronal and the glial populations, that it would be possible to do those assays in vitro.
Sure. Okay, now it's expert, glial expert. Yeah, please.
Yeah.
Thanks. Hi, Corey. Really great work.
Thank you.
I'm just curious about the spark part, because spark has previously been shown to inhibit synapse formation. Do you think it's context dependent, or depends on how the experiment was done? There are explanations for the discrepancies.
Yeah, so we think that it's probably a context dependent difference in sonic. So, and that's actually also seen with spark, where there's previous data showing that spark had a role in promoting synapses in hippocampus, whereas in the cortex it seemed to be less clear, I think.
How do you think the dependence on hedgehog interacts with the well-known patterning rules of hedgehog and brain development?
Sorry.
Good to see you, Lou.
Sorry, I just wanted to ask. I mean, since hedgehog has got some, sonic hedgehog in particular has got such well-known roles in patterning the brain.
Yeah, I think that one of the things that we're most curious about is how is it that the hedgehog is actually being delivered by neurons. So I think that it's an interesting question, and with respect to what we see in the layers, where again, if we look at where GLI1 activation is highest, it's in proximity to the cell bodies of those neurons. But many of the effects from the loss of function we see throughout the layers of the cortex, which might suggest that there is some kind of gradient of hedgehog signal found throughout the cortex, where it's highest in the deep layers. And then, so in that way, I think what we've learned about sonic hedgehog signaling and patterning embryos might teach us something about what it's doing in the cortex, or how different levels of the sonic signal might provide kind of different contextual cues.
Okay, thank you.
Thanks, Lou.
Yeah, I think, did I?
Okay, Shinjae, please. Yeah, go ahead.
Corey. Hi, how are you?
Hi.
Shinjae from UC Merced. Okay, so just a quick question. So you have the tool to look at the patched receptor expression in the brain, the cortex. Have you looked at the developing cortex at the stage of neurogenesis and where patched are expressed? Because the field believes that the patched receptor expression during neurogenesis is very low, right? So have you looked at that?
Yeah, so actually, and so I think at least with respect to the cortex, so we looked prior to P5, I can't remember if it was late embryonic, but we've definitely looked at, say, P0, and you don't see patched expression in the cortex. You see it, in places where you might expect in the proliferative zones, like in the subventricular zone, for instance, but as far as in the cortex and the gray matter, you actually don't see patch expression until that later postnatal period. But we haven't looked all the way back towards earlier parts of neurogenesis.
I see. Are you planning to see that?
I think we're probably, for at least with respect to this signal, looking mostly in the postnatal cortex.
I see. Okay. All right. Yeah. Thank you.
Hey, Corey, that was great talk. It's cool to see these early developmental genes play functions in such a later stage, and presumably by activating different sets of genes that are, in this case for synapse formation. And I have two questions. One is, have you knocked out smoothened or glee in astrocyte to see if you remove the hedgehog signaling as opposed to activating? Do you see the reduction of synapse formation, and how much do you see the reduction? The other question is you saw a higher expression of hedgehog actually patched in neurons at P60, much later. So any speculation about what hedgehog signaling is doing in neurons at the more mature brain at that time?
Yeah. So I think to the first question, we've looked a bit at gene expression from the smoothen knockout. And there's work previously from Denise Garcia's lab where they've knocked out smoothen. And actually they see an increase in excitability of those neurons. And I think also an increase in synapse. So it is a little bit of the kind of opposite observation, but actually I think that it's probably because of a distinctive mechanism of the function, because all of these genes are being misregulated, and especially for the neurons, it could be that the loss of smoothen keeps the astrocytes from receiving the sonic signal. But from our previous work, we know that there's still neurons that are expressing Bach that could still respond to sonic hedgehog signal. So, how is it that, the loss of the ability to respond to the sonic from one cell population impacts the other, I think is unclear. We're actually really interested to kind of try to figure out how those things might change using our system. And then for the next question, the function of patched in mature neurons, actually we have no idea what it's doing. So what we do know is that if we were to look at, say, Glee family transcription factor expression, we never see that in neurons. That stays exclusive to the glial cells. So how is it that patched might be signaling within neurons is still a mystery to us.
That was very cool, very cool. Yeah. It's possible that in the later stage, the signaling mechanism might be a little different. Not necessarily exactly the same.
Right. Yeah. I think that's exciting for us, especially to think about whether there's some difference in signaling, say, in the cells that would co-express both Bach and patched, the neurons, I mean, versus the neurons that are exclusively expressing Bach. So how might that also kind of add an additional level of how the cell's response to sonic might be altered?
So I guess there is a precedent and it acts on guidance, right?
Yeah.
Any more questions? Okay, no more questions. Thanks, Shinjae and Corey for beautiful work today. Okay, thank you, everybody. See you all next week. Another, hopefully, still quarantine or spread out your Zoom in Shanghai.
Thanks,
Corey.
Shinjae.
Thanks, Corey. Thanks,
Thank you, guys.
Beautiful work.
Bye.
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 See you all next
Bye,
week. Bye.
Bye, everyone.